Okay, well, thanks for that last talk. That was a great talk from my colleague, and many of the things that you saw in this, particularly in those last few slides about CAR Ts, issues that firmly fit into this particular topic. So, I guess this is a question of whether you're a buyer or a seller. If you're selling the products, we've got no end of opportunities for people to sell products. But my position here is really one of a buyer, and that's the fundamental difference between, I think, people who are selling hope and us who have to buy value for the populations that we serve. So again, I just wanted to start again with this little diagram, which is really around how do we ensure that we're buying health outcomes at the time that we are sold the product, and continuing to extract value from that product during the life cycle of that particular product. And it's not a matter of setting and forgetting. We've got the product, let's just not pay any more attention to it. But we know that we do need to continually keep an eye on whether we're still extracting value from these products. It's long been known that the initial results seen in clinical studies are often overestimating the effect size of therapeutics and underestimating some of the downsides. And we know that the recent review that the FDA did on their accelerated access showed that many of those products actually didn't live up to what they had originally purported to deliver for the population. So we can never be complacent about thinking, well, we've made an assessment, we're really buying something of high value. We need to constantly be vigilant about these products, particularly when we look at the price tag of these. So we need to have a very proactive healthcare system. The healthcare system is often very slow to move, and yet we've got this very dynamic industry that is selling us products. How do we make sure that we get a nice match between the seller's motivation and also the needs of our community? So as we all know, being a smart purchaser isn't actually always that easy. It's very challenging, and HTA methodology is not the golden solution to all of these problems. Just applying methodologies from a toolkit doesn't necessarily mean you're a smart buyer. And particularly when we're looking at health technology tools that were developed a long time ago and have been in many ways very slow to catch up with the changes in the environment in which we're actually buying technologies. So we need to constantly update these technologies, constantly be thinking, but who has time to think when all of these things are coming down the pipeline all the time and people are busy trying to do their work? It's hard to sit back and think, well, have we really got the right toolkit for evaluating these things? Advocacy groups, I mentioned in my last talk, advocacy groups are increasingly organised in all countries, and they have very specific expectations about how healthcare financing should be done, which services should be funded, and how they think their prioritisation list should go. But do they really reflect the values of our community as a whole? As we've heard from the previous talk, the pace of technologies are increasing at a great rate, so it's very hard to keep up. And managing the approval and reimbursement decisions in an efficient way is an ongoing challenge for every country. And finally, as I said, just sort of having that whole of life cycle assessment is very tricky. Okay, so what I wanted to do was actually just touch on 3 different examples of things that we're facing in Australia to give you a sense of the complexity of what's being assessed when we're looking at, are we selling hope here for cancer or really genuine outcomes. I'm going to talk about Theranostics as an example of something we've recently had to start facing into, cancer genomics and CAR Ts and biospecifics. I'm just going to mention at the end because you've heard about that and we have a session later about that particular topic. So we have an ever-increasing array of new technologies. The CAR T treatments are coming thick and fast. In the beginning, they were coming for rare diseases like acute lymphoblastic leukemia in children, so it's much easier to consider that. But now we're seeing more and more of these CAR Ts put through for diseases like myeloma, where there is a lot of other treatments available. Where do CAR Ts fit in that paradigm where we're treating a lot more patients with chronic disease who are much more elderly, who have a lot of other treatment choices? It's not the same as a small group of children with ALL that is refractory to all treatments. We've got cancer vaccines coming around for GBM and mesothelioma. Other transplant therapies, T-cell therapy, a lot of different therapies now, and the big one at the bottom is this increasing use of T-cell directed bispecific antibodies. So these T-cell redirecting bispecific antibodies are a whole new paradigm of treatment, and we're going to see more and more of these therapies on our doorstep very soon. Okay, so let me talk now about these three topics I want to particularly cover. The first one is Theranostics. And this is the use of a diagnostic compound and a therapeutic compound so closely together that we decided to invent a new word for it called Theranostics. And Theranostics doesn't always involve the same radioisotope as is bound to the same ligand, and I'll explain what that means in a moment. But Theranostics actually isn't a new topic. It's been around for a very long time, but companies are now very interested in selling these particular products for cancer. So we know that people have treated patients with malignant thyroid cancer, metastatic disease for a very long time with radioisotopes, the radioiodine, and that's been used very effectively for many decades. And in fact, it is actually a theranostic. It actually targets, it uses technetium scans to identify the target, then designs the treatment and administers a therapeutic dose of radioactive iodine to eliminate metastatic disease almost in about 90% of people. So it's really fundamentally is a theranostics, but never labelled so. We also know that we have lutate for the treatment of NET tumours. This has also been around for a long time and is a highly effective treatment for metastatic disease in patients with NET tumours. It's another example of a theranostic. So why is this becoming a bit more contentious than previously? And I'll just go through a specific example that we've encountered. This is PSMA targeted radionucleotide targeted therapy. This is precision radiation oncology. And it's targeting a cell surface glycoprotein. 95% of the molecule is situated on the extracellular membrane. So it's a fantastic target, very effective target for prostate cancer because it's highly overexpressed on the prostate cancer cells and metastatic prostate cancer and isn't expressed much on any normal cell. So it's ideal as a therapeutic target. So PSMA is very easily accessible, and the easiest way to assess it or to access it is to develop small ligands, and that's the little red blob there, the PSMA ligand. That's a small molecule which will easily go and bind to PSMA. All you need to do is to attach to that red blob a radioisotope. If you attach a radioisotope, then you've got a really strong compound that is actually going to target metastatic prostate cancer and be highly effective at killing the prostate cancer. So there are in fact a couple of different ligands, and this is where it gets a bit tricky. So those red blobs exist there's two of them at least and many more. One of them is called PSMA 617 and another one is PSMA IT. So a pharma company has undertaken what you would expect in metastatic prostate cancer. They've done a randomised control study looking at the use of radioisotope targeted PSMA 617, so not IT, 617. So the red blob here is the PSMA 617 and they've attached a very effective beta emitter, lutate 177. And this study showed convincingly that patients with metastatic prostate cancer had improved quality of life, a lot less pain, and increased survival compared with standard of care. So this looks like a highly effective therapy. It was published in the New England Journal of Medicine in 2021. So then it gets a little tricky because the question is lutate PSMA IT the same as lutate PSMA 617, because there are other red blob molecules, and should we treat them as exactly the same? The therapeutic agent is actually the radionucleotide, not the binding molecule, not the ligand. But where does the IP, intellectual property, actually exist? Is that red blob really where the essence of this therapeutic effectiveness lying? And so they are exactly the same class of molecules. They're both small molecules inhibitors. They're both targeting PSMA. They've got a very related chemical structure, but it's not identical to each other. They're using the same beta emitter to treat patients, and they have similar preclinical assessment in a mouse. So this isn't our traditional HTA space. High specific binding, IC50 values, comparable tumour uptake, etc. So we might expect them to behave similarly, but can we assume that they have comparable efficacy and toxicity? Could we assume that, or can we not assume that? And one's a patented compound and one isn't. So these are empirical questions that we've not previously faced in an HTA context. Can we assume a claim of non-inferiority can be established in this case? And what methods do we actually use to establish those two compounds as equal? Do we use molecular kinetics? We've never done that before in HTA. Do we use measured radioisotope dosing? Is the measured radioisotope dose different in different prostate cancer mets? Is each agent non-inferior in patients with metastatic cancer, castrate-resistant prostate cancer? Do these new agents have to do the same reproducible phase 3 clinical trial, or do we treat them in a way like a Me Too drug? And this is a whole new area that's blooming at the moment, and I think something we're going to see more and more of. So, second topic was about cancer genomics, and you could talk on this topic for hours, but I thought I'd just mention a couple of examples. The first one is gene expression profiling for predicting chemotherapy benefit in breast cancer. This is using expression profiling using mRNA, using multi-gene signatures, 70 genes at a time assessed. There's multiple products that do this. And secondly, I'll just touch on in the last few minutes somatic gene testing for diagnostic utility. So what has become very confused in this area is the difference between prognostic value and predictive value, and I guess we all learned that when we're sort of studying HTA 101, but it is actually increasingly important that people understand the difference between prognostic value, which is essentially providing a marker of the natural history of a disease, versus prediction, where the marker actually predicts whether there's a differential effect of that treatment in the people who have the biomarker versus those who do not. And it's an important distinction. So the current treatment for breast cancer is that patients have their surgery, they have their clinical assessment, and then they have either chemotherapy or no chemotherapy. And the idea here with this particular expression profile test was to introduce something called MammaPrint, and there are others, Oncotype DX, ProSigna, EndoPredict, there's lots of these. Introduce that test after the person has had the clinical assessment and say, well, we're now going to recategorise you as either a high risk or a low risk. And the idea here is that genomic testing would be a chemotherapy sparing test. So in essence, the argument is that QALYs are being saved because patients are avoiding unnecessary chemotherapy. And we know that there's an absolute large number of women who received chemotherapy post-diagnosis of breast cancer who didn't really need it because their survival was going to be guaranteed one way or the other, but they're having it more as an insurance treatment. So if we look here, this study was done was to try and look at patients who were classified as high clinical risk, but when the MammaPrint test was done, they were recategorised as low genomic risk and therefore could safely avoid chemotherapy. And that's the argument about these gene expression tests. Now, in assessing non-inferiority for a genomic test, I think this is a very useful paradigm. If the MammaPrint decisions are better, so essentially what we're looking at is, can we safely avoid a treatment? In other words, can we assure ourselves that in every clinical parameter that we're measuring, we've not crossed over that non-inferiority margin? And to do that, it's a nice little schema here to look at that. If non-inferiority is shown, you'll see your confidence intervals and your point estimates up on that right-hand side. If non-inferiority is questionable, you'll see the lower bounds of the confidence interval will cross that non-inferiority threshold. But if all your analysis is over in the other end, you're probably buying an inferior product. And this is what the MINDAC study, which looked at the value of MammaPrint, showed. In every instance, overall survival, disease-free metastatic survival, disease-free survival, all of the point estimates and all of the confidence intervals were all on the wrong side of that non-inferiority threshold. In other words, this was selling hope. It wasn't selling something that was guaranteeing that chemotherapy could be safely avoided. And as you all know, a principle in medicine is to do no harm. And what we did in the MSAC assessment of this is just looked at a population level, how many women would end up with having a local recurrence, having disease that they died of, or having metastatic disease and having to live with metastatic disease as a consequence of introducing this at a population level to every woman with early stage breast cancer. And on a population level, the decrement in survival was roughly 2 or 3%, which is the same increment in survival we were buying with adjuvant chemotherapy in the first place. So it's an example of where, I'm not saying every one of these gene expression profiles has this same issue, but this is an example of why careful analysis needs to be done. So in the final couple of slides, I just wanted to mention a couple of other ways in which somatic diagnostic gene testing is being used. It's being used to replace the disease classification systems that we currently have in place in oncology. So in other words, it's becoming another way of classifying tumours in addition to the morphology or the way the tumour looks under the microscope. And this is creating issues for us in terms of how do we have a reference standard against which we define a classification? And many of these diagnostic tests are now having to refer back to the WHO classification of using molecular testing to classify tumours. But that's a very big task to keep that information up to date. And yet I don't think it's anything that a single country can do. And we really do need to collaborate across many different countries in order to be able to keep our diagnostic disease classifications up to date, because that's the fundamental basis on which we're going to fundamentally decide on different treatments. Secondary purpose, apart from diagnosing disease and accurately classifying it, is, as I mentioned earlier, about prognosis. Does this molecular test tell us whether patients are likely to have a better natural history or a worse natural history? Very important purpose. And then a tertiary purpose that is increasingly being used is for the access for patients to clinical trials. So if you need a particular molecular profile in order to get into a clinical trial, well, that molecular profiling done at this particular diagnosis of cancer is useful for generating more evidence about the value of different therapies. So these sort of integrated diagnostic tests are really challenging to work out how you assess their value in a health technology assessment sense. As I said, it's very difficult if you don't have an international reference standard against which you can compare your results. How big should these panels be? Some of them, should they have diagnostic, prognostic and predictive purposes, or just one or the other? Is that enough? As I mentioned earlier, this may be optimal panels for diagnostic utility may actually differ between the diagnostic pathologist and the treating clinician. The treating clinician might want this, the pathologist might say, I only want these pieces of information. So we're seeing these interprofessional disagreements between what both different sets of doctors actually want. And then there is variability in the composition and panel testing of different providers. So how do we have a quality system that ensures that each patient having a test is actually getting the right test for them and it's giving them the right information, and they wouldn't have got a different result if they'd had that test somewhere else? So there's been a number of attempts now to try and look at how we would classify different variants. I'm not expecting you to read this because it's something that is in evolution, but it's part of the framework that we're looking at when we're trying to assess the value of new tests, and then how we connect that up with therapies. Is these frameworks are coming out all the time about when can we really be confident there is strong clinical evidence that that particular piece of genetic information has enough veracity for us to then base a treatment on it, or when is it much weaker evidence? And we're so reliant now on colleagues from around the world to help push these different pieces of information into a framework which is constantly changing. So I didn't want to leave you depressed after that long talk, which sounded like, oh my goodness, that's an overwhelming set of things to think about. But I always choose to be optimistic because it feels a lot better. Thank you very much.